My name is Sven Tozer. I'm from Rhodes University. I'm an honours student working under Martin Hill and Julie Kutsia. And today I'm going to chat to you about one of my honours projects, looking at the relationship between food quality and fecundity, as well as the wing form in Megamelus scutellaris. Okay. Megamelus is a sap-sucking plant hopper from the family of the Delphasidae, and it's host-specific to water hyacinth and originates in South America. The species exhibits what's called wing polymorphism, where it'll produce a fully winged macropterous form capable of dispersal and a reduced wing brachypterous form, which is mainly sedentary. And from the literature, it seems that there might be an energetic trade-off between two different wing forms in different types of situations, with the brachypterous form being a lot larger and more fecund than the smaller macropterous form. So my project has a number of core questions which we need to answer in order to understand this mechanism. Firstly, does nutrition have an impact on the production of egg and sperm within this species? And how does this differ between the two wing forms that we get? Thirdly, does this differ between the nutrient treatments between the wing forms? And lastly, is there an effect of maybe population density affecting the wing form ratios that come out of that? My hypothesis is twofold. In terms of nutrition, in stable densities, the nutritional quality of the host plant or the nutritional status of the water body is largely going to determine the ratio of the two wing forms. And in terms of density, if we keep our nutrients stable, we should see a shift in the wing form ratio from a macropterous or brachypterous form or in between. And I will elaborate on that in a moment. So my methods have changed as Grahamstown gets pretty cold in winter. But essentially I have three nutrient treatments where I've introduced nitrogen, phosphates, and obviously iron chelate in a low, a medium, and a high concentration. And from there we measured basic plant parameters that have been set out in a number of other studies: PTL length, the root mass, leaf mass, etc., etc. And to do this, we actually did a study outdoors, but it was evident that winter was going to hamper the results, so we moved it indoors, and we created a microcosm inside, growing water hyacinth in a circulated hydroponic system, which allows us to control for light, pH, temperature, water circulation, and allows us to monitor these plants in a more controlled fashion. We'll then introduce insects onto these plants, and from there, we are going to test the potential fecundity through dissection of the seminal vesicle, the vas deferens, the testes, and the ejaculatory duct. In this picture over here, you can see those are the testes over there. And there we go. And this is actually a really closely related species, Peregrinus matus. It's within the same tribe, though there's been no work done on this species, so we're just trying this out for the meantime. From there, we're going to stand with the hooks and illuminate that and take digital photography and count the total number of sperm heads per sample. For our females, we're going to do a pretty similar technique, but we just dissect out the ovaries and the ovarioles and count the number of eggs inside there. In order to quantify this in a more of a viability study, we're going to do oviposition trials. We're counting obviously the number of eggs per clutch and the number of eggs which have hatched from that clutch. In order to analyze all of this, we'll just compare the counts between the wing forms, the macropterous and brachypterous forms, and compare that across nutrient levels. In conclusion, releases of control agents should be targeted to specific requirements, and that has been highlighted in a number of talks over the past 2 days. Control agent efficacy and establishment probability may be highly dependent on the number of different regional nutritional status of water bodies. And if this study does show that nutrients have a high degree of influence on the fecundity of Megamelus, then study sites should be chosen that favor the establishment of the relevant wing form in order to increase their efficacy.